Hi students, this is A. Mandari Kumar Rajesh. In this video, we are going to discuss another major component of gene cloning experiment called vector. What is called vector? What is the role of vector in gene cloning experiment? Vector can be defined as a DNA molecule which can carry the gene of interest into the host cell, which can transfer the gene of interest into the host cell, and it has ability to self-replicate inside the host cell. Then only we are able to get multiple copies of gene of interest or recombinant DNA. So in recombinant DNA technology, we have to isolate a plasmid. So this is a plasmid or a vector. Then we have to isolate the gene of interest. So the gene of interest has to insert into the vector by the process called insertion. For that process, we need two types of enzymes. They are restriction endonuclease, then DNA ligase. So with the help of restriction endonuclease, we can cleave the vector at the specific region. We can cleave vector on the specific region. Then with the help of the DNA ligase, we can ligate the gene of interest with the vector. So with the help of DNA ligase, we can ligate the gene of interest with the vector. So that process is called as recombinant DNA technology. Then this recombinant DNA inserted into the host cell for multiplication. So along with the host cell, the gene of interest also get multiplied. Then we are able to get more number of gene of interest or recombinant DNA. So the vectors are otherwise called as cloning vehicle or cloning DNA. That is why the vectors are otherwise called as cloning vehicle or cloning DNA. So a vector can be defined as a DNA molecule which can carry a foreign gene or gene of interest into the host cell, and it can self-replicate inside the host cell for getting more number of recombinant DNA or gene of interest. Then the vector may be a plasmid, extra chromosomal material which is present in the bacteria. Then it may be a cosmid, then it may be a pagemid, then bacterial artificial chromosome, or it may be yeast artificial chromosome, or it may be a transposon or jumping genes, or it may be even a virus. So these are all some of the types of vectors which are used in gene cloning experiment. Among this plasmid, and then one more I have missed, bacteriophage. Another vector is called bacteriophage. So the most common used vectors are plasmid and bacteriophage. So the most commonly used vectors are plasmid and bacteriophage. Then vector can be classified into two types based on their mode of action or function. Let me tell about the two types of vectors based on their function. They are cloning vector. The second one is expression vector. So I give you the definition and the difference between these two vectors. So the cloning vector can be defined as it is a DNA molecule which can carry the gene of interest or the foreign DNA into the host cell, and it can self-replicate inside the host cell, and that vector is called as cloning vector. While in the case of expression vector, the expression vector is a plasmid, not only transfer the gene of interest, but it also helpful in the analysis of the gene of interest through relevant protein product expression. That means this expression vector can produce a protein related to the gene of interest. So the gene of interest is nothing but a DNA. So the DNA must to transcribed into mRNA, and that mRNA translated into a protein. So that is called expression vector, or it is otherwise called as expression construct. Expression construct. That means that expression vector contains the operator gene, the promoter sequence, then transcription initiation sequence, then transcription termination sequence, the ribosome binding site, then gene encoding repressor that binding O and regulates promoter site. That means it contains the promoter site, then transcription initiation sequence, and it contains transcription termination sequence, and it contains translation site. So these all the things tell us it commands the gene of interest to produce a protein. So this setup, this construction commands the gene of interest. Gene of interest is nothing but a DNA molecule. Command the gene of interest to produce a protein molecule by the process transcription and translation. That is called central dogma of molecular biology. So that construction demands the gene of interest to produce a protein molecule, and that type of vector is called as expression vector, and that type of vector is called as expression vector. It expressed by the production of protein. That is why it is called expression vector or expression construct. Expression construct. Then the cloning vectors are used to obtain multiple copies of gene of interest or foreign DNA, while in the case of expression vector is used to obtain or analysis the gene product. In the case of expression vector, it is used to obtain or analysis the gene of interest. Then the cloning vector may be a plasmid or cosmid or a pagemid or bacterial artificial chromosome or yeast artificial chromosome, and it may be a virus. While in the case of expression vector, it must be a plasmid or a virus. In the case of expression vector, it must be a plasmid or a virus. Then in the case of cloning vector, it contains ORI site, that is the origin of replication site, and it contains selectable marker. Then it contains restriction site or a specific site where the gene of interest get ligated. So the cloning vector contains origin of replication site, then selectable marker, and the restriction site. While in the case of expression vector, it contains the promoter site, then the operator sequence, then transcription initiation sequence, and it contains transcription termination sequence and translation initiation site. In addition, it contains origin of replication site and selectable marker and the specific site or recognition site. So it contains transcription initiation sequence and transcription termination sequence, then operator site, then promoter site, then ribosome binding site, then gene encoding repressor binds operator and regulates promoter. In addition to that, it contains origin of replication site, then selectable marker, and specific site or recognition site. So that's all about the difference between cloning vector and expression vector. Now I am going to tell about the properties of a good vector. Let me explain the properties of a good vector. Here I have written 6 points about the properties of a good vector. Then I will explain each and every point one by one. The first point tells us vector should be small and easy to isolate. Then only the vector can easily transfer into the host cell. So the vector should be small and easy to isolate. Then only it can be easily transferred into the host cell. Then vector must contain one or more origin of replication sites so that it can stably, independently replication inside the host cell. So a good vector must contain at least one origin of replication site. So this is the origin of replication site. So in the origin of replication site only the replication process get started. So the vector should contain the origin of replication site. Then it should contain replication protein. So the replication process is initiated by the protein called replication protein. For example, in DNA replication, the replication process is initiated by a protein or a primer or a RNA. So that primer can initiate that replication process. Then only the DNA polymerase can recognize and binds with that primer and it starts it initiates the DNA replication process. Likewise, in vector also it should contain the replication protein. Then only the DNA polymerase can recognize and bind with the replication protein and it can initiate the replication process. So the vector should contain the replication protein. Then the vector should have one or more unique restriction sites into which the recombinant DNA can be inserted, because if the vectors contains there is multiple cloning site or polylinker site, it facilitates the use of restriction enzyme of choice. That means, so this is a structure of cloning vector. It contains multiple cloning sites. It contains multiple cloning sites. That means it contains multiple restriction sites or specific sites. So we can insert the gene of interest in any stretched gene sequence. So we can use a restriction enzyme ECODE 1, and we can insert that gene of interest in that site. Otherwise, we can use HIN3 restriction endonuclease enzyme, and we can make a cut and we can insert the gene of interest on the specific site. So we are able to use any kind of restriction endonuclease of our own. So we have many number of choices. We can use any kind of restriction endonuclease because of the presence of multiple cloning sites or polylinker sites. Suppose if it contains only one cloning site, so we have to use specific restriction endonuclease. For example, if it contains ECODE 1, we have to use only ECODE 1. But in this case, we have multiple cloning sites. We can use any kind of restriction endonuclease to get inserted the gene of interest into the vector. Then it should contain a selectable marker such as antibiotic resistant gene. So commonly it may be a antibiotic resistant gene to permit a detection in transform host cell. That means it contains a selectable marker and it is an antibiotic resistant gene. It is a antibiotic resistant gene. So during recombinant DNA technology, we have to isolate a plasmid, and that plasmid get cleaved by the restriction endonuclease, and the gene of interest get inserted with the help of DNA ligase. So These are all plasmid get cleaved by restriction endonuclease. Then the gene of interest get ligated with the help of DNA ligase. At that time, some plasmids get self-ligated. Some plasmids get self-ligated. But we need to isolate only the transformed cells. We need to isolate only the transformed cells. That means the recombinant DNA. Then how do you isolate the transformed cells? How do you isolate the transformed cells? So for that purpose we need a selectable marker. For that purpose we need a selectable marker. Usually that selectable marker is an antibiotic resistant gene. Suppose that antibiotic resistant gene is a streptomycin. So that antibiotic resistant gene resistant to the antibiotic streptomycin. If we place these plasmids or these vectors in the antibiotic streptomycin, the plasmids which contains that selectable marker which can resist to the antibiotic streptomycin. So these are all. So these two are recombinant DNA which contains the recombinant DNA. That plasmids contains recombinant DNA, and we have to place these plasmids into the antibiotic streptomycins. So these two plasmids which contains recombinant DNA shows resistant to the antibiotic. Others may not be grown in that culture, So with that we can isolate the transformed cells. For that purpose we need selectable marker. So it should contain a selectable marker such as antibiotic resistant gene to permit its detection in the transformed host cells. So with that selectable marker we can isolate the recombinant DNA, that is the plasmid with the recombinant DNA. Then the vector should not be a toxic to the host cell. Finally, the vector should not be a toxic to the host cell. Otherwise, the host cell may not able to multiply the gene of interest. If the vector affects the host cell may not be able to multiply the gene of interest. So that's all about the vector. If you have any doubt, please let me know. Thank you very much for watching this video. Thank you.